What are genome editing and CRISPR-Cas9? Genome editing, also called gene editing, is a group of technologies that give scientists the ability to change an organism's DNA. These technologies allow genetic material to be added, removed, or altered at particular locations in the genome. Several approaches to genome editing have been developed. A recent one is known as CRISPR-Cas9, which is kind of like short for clustered, regularly interspaced short palindromic repeats, and CRISPR-associated protein 9. The CRISPR-Cas9 system has generated a lot of interest in the scientific community as it's faster, cheaper, more accurate, and more efficient than other existing genome editing methods today. CRISPR-Cas9 was adapted from a naturally occurring genome editing system in bacteria. The bacteria capture snippets of DNA from invading viruses and use them to create DNA segments known as CRISPR arrays. The CRISPR arrays allow the bacteria to remember the viruses. If the viruses attack again, secondary infection for example, the bacteria produce RNA segments from the CRISPR arrays to target the virus's DNA. The bacteria then use Cas9 or a similar enzyme to cut the DNA apart, which pretty much renders the virus ineffective. The CRISPR-Cas9 system works similarly in a laboratory, where researchers create a small piece of RNA with a short guide sequence that attaches to a specific target sequence of DNA in a genome. The RNA also binds to the Cas9 enzyme. As in bacteria, the modified RNA is used to recognize the DNA sequence, and the Cas9 enzyme cuts the DNA at the targeted location. Although Cas9 is the enzyme that is used most often, there are other enzymes, of course, that can also be used. Once the DNA is cut, researchers use the cell's own DNA repair machinery to add or delete pieces of genetic material, or to make changes to the DNA by replacing an existing segment with a customized DNA sequence. Genome editing is of high interest in the prevention and treatment of several human diseases. Currently, most research on genome editing is done to understand diseases using cells and animal models. Scientists are pretty much still working to determine whether this approach is a safe and effective method in people. It is being explored in research in a wide variety of diseases, including single gene disorders, for example, hemophilia and sickle cell disease. It also holds promise for the treatment and prevention of more complicated diseases, for example, cancer, mental illness, and one of the big ones, human immunodeficiency virus, HIV. Ethical concerns arise when genome editing uses technologies, CRISPR-Cas9, to alter human genomes. Most of the changes introduced with genome editing are limited to somatic cells or body cells, which are cells other than the egg and sperm cells. These changes affect only certain tissues and are not passed from one generation to the next. However, changes made to genes in egg or sperm cells, known as germline cells, or in the genes of an embryo, could be passed to future generations. Germline cell and embryo genome editing bring up a host of ethical challenges, including whether it would be permissible to use this technology to enhance normal human traits, for example, height, eye color, even intelligence. Based on these ethical concerns, the safety of CRISPR-Cas9 has come into question, and of course, its use in a laboratory today raises many concerns of commercialization and where it's heading in terms of what it should be applied to.